Alrighty, so as I said, I'm going to be talking about use of, hey Rob, ultraviolet lights and molecular diagnostics. It is really a cast of quite a few people who are working on this. This is a collaboration between 3 of our Crown Research Institutes, Scion, Lancaster Research, Plant and Food, and also Wilderlab, who are New Zealand's only commercial eDNA laboratory, which they may be familiar to many of you. Okay, so the issue, why are we doing this particular research? And I guess for many people in the audience, you know what that's about. But if we want to be a trading nation, there's always going to be biosecurity risk. There's no such thing as zero risk when it comes to biosecurity. We bring in millions of shipping containers per year, so the odds are better than lotto that something's going to happen. You know, 1 in a million chances are happening every year. Okay. And there's this big diversity of things that we know that are a threat to us, things like brown mother, stink bug, spotted lanternfly, xylella. You know, we know about certain high priority threats, and we know that because they cause damage elsewhere. But we also know that there's all these unknown things that we don't know about. And if we want to be able to detect those unknown things early, such that we might be able to eradicate them after they arrive, we need to have some form of large-scale sampling. And I know there's quite a few people in the audience, both online and here in person, who, you know, their job is around finding those things early. And you know how intensive that is in terms of staff time to get around places. But then there's also the diagnostic impediment of, okay, if you've got a big, you know, set of samples of something, someone has to go through them and find that needle in the haystack. Because ideally, if you're finding something early, it is the needle in the haystack because they will be at low abundance at that point in time. So, sorry, I had forgotten that was in there. I should have taken this slide out. I gave this to an international audience. So we have these various surveillance programs that are out there, things like high-risk site surveillance, etc. And the idea behind these tools is not to replace these surveillance tools that we already have, but to augment them and make them stronger and better. Okay, and so many of you will be familiar with this high-risk site surveillance, where we're going around doing transects annually. These transects are set aside as to where they're going to be based on where volumes of imported goods are coming and where there are areas of rich vegetation nearby those. But you can't survey all of those areas in there. So perhaps if we can get tools that help us get a broad brush across all of those areas, we can help target the inspector effort to go and look at particularly suspicious things. So the research program is in 2 halves, and today we've got the 2 talks. So first of all, I will be talking about the use of DNA to detect signs of unwanted organisms. So a lot of our surveillance works on signs and symptoms. So signs are being the physical, actual presence of the organism. You have its frass, you have the organism, you have a fungal fruiting body, you actually have a piece of the biological material of the organism. Whereas symptoms are the effect that they cause in the tree. So the tree might be wilting, there might be branch dieback, there might be things going on, okay? And where we're looking for symptoms, that is what Paul is going to talk about, is about using computer vision to be able to decide if trees are going from a state of healthy to unhealthy, so that we can target inspector effort to go and check those out. So starting off with the DNA thing, DNA is a very powerful tool. We're all leaving fragments of DNA everywhere. So I can guarantee you that if we did a forensic analysis of this room, we could probably detect that all of the people in this room were in the room. You know, you would be able to come in and swab everyone and you would be able to do it. So we're all shedding skin and stuff and it's all going everywhere. But the question is, if we want to use these for surveillance, how do we aggregate this in a useful way that we can find things early? And people have used these, and you've probably heard some of the headline cases of things like scraping stuff off license plates, but you can also use vacuum cleaners. The Australians are doing a project around shipping containers and vacuuming out the floors of shipping containers to understand what might have been in that shipping container, to understand what is on those pathways. Here in New Zealand, there's been a great amount of work done by the Cawthron Institute, looking at rolling out these DNA-based tools in the marine environment. And there's some tools available there through Cawthron's website. But we're looking at the terrestrial environment here, and we're wanting to find things early. So we're focused on UV light traps. And the reason for focusing on UV light traps is that for flying insects, UV light is a fairly conserved feature of the insect vision system, such that most, not all, but most things that are attracted to light will be attracted to UV light. So prior to us starting with this, when I was at Scion, Carl and myself, when I used to be at Scion, we did some work with these pretty rudimentary light traps. As part of the project, we've developed a more comprehensive light trap that is much more robust. They are more difficult to put up. They're designed to fit on these kind of security gate posts because most transitional facilities or ports do have some of these in their environments, and they're designed to be, put them up and leave them there somewhat permanently, hence they're a bit more robust. They have a solar panel to allow us to charge the batteries, and they have, can't see in this image here because it's behind the solar panel, there's a little antenna, which is what this cable goes to. So we can interface with that with the communication systems. You may have heard of Internet of Things communication. So we can give it simple instructions to say, here's a schedule of when we want the light on or off. So we can change it anywhere pretty much at any time. And then an inspector would have to come along and change out the traps, and then the trap contents will be taken to the lab for processing. So this is the element of getting this together, is to have a robust tool that we can put out there and leave out there, and then periodically when we want to, we can take samples by bringing a sample collection jar with some collecting fluid in it, and then coming back a couple of days later to take the samples away. And we take them away to a lab. And so this is the rough sort of element of what we're doing in the lab. For many people, you can probably switch off because you'll know all of this, but for those that don't, so we'll take our samples, and we've done some test work with this at the Port of Tauranga, POT, so we've got these samples, and we want to sieve them. And the reason for sieving them is if you have a couple of individuals in your trap that are making up 95% of the biomass, when you extract the DNA, 95% of the DNA that you're likely to extract has come from these two individuals. And so when you do your analyses down below, your further analytical steps, you may saturate your reactions with the DNA from those large individuals. So by sieving them into different groups, we can get better representation. So we're extracting the DNA, and we're using a process whereby we're not having to blend up the insect into a pulp to get the DNA. We're trying to elute that DNA out of the organism whilst keeping the organism whole, which is important for verification purposes. If we get a hit on something, we can go back and have a look in those samples and go, yes, there was one of those in there. Okay, so we've been trying it with a number of primers. There's one underneath the screen here, I think. So 1, 2, 3, 4, 5 different primers. The idea of having different primers is some of them are better at amplifying DNA from different groups. You can't see it under the screen here, but there's a little brown marmorated stink bug down in there. And that's because this sterno primer that's in under here is particularly good at targeting Hemiptera, of which brown marmorated stink bug is a Hemiptera. Okay, so we've got this process of collecting the samples. Our aggregation method is the UV light, extracting the DNA, amplifying up small sections or barcode regions of DNA, and then sending them off for sequencing. And then the nutty bit happens around then, well, how do we do the bioinformatics of that? We've got millions of sequences. How do we assign those to species names to figure out if we potentially have a new to New Zealand thing that we need to be worried about? And so the crux of all of this is the reference database. And so you can think of reference databases. This is a wonderful little slide that Sean from Augalab shared with me to use, and it really gets across really well. It's exactly the same process that you're going by with fingerprint matching for a crime scene. You can easily get fingerprints from everywhere. We've all left our fingerprints on the doors that we came into these buildings, but we can't say it's you unless we already have your thumbprint from somewhere to match it with. It's the same principle with a reference database. So we have put together a reference database of various things, which I'll go to in a minute. So from the Port of Tauranga, I'm going to say we had the 40 odd samples that we did. We had them at these different areas within the port. So we had some in the log yard, we had some by berth number 2, some in the container terminal, etc. And we sampled them over 3 different weeks. And these are the number of what we call operational taxonomic units per sample. So we've used a threshold as to how different the sequences need to be considered to be a different species, but we haven't yet put any names on those species. Okay, so this is for one gene region. I think from memory it was around about 1600 different taxa that we found. And it's not surprising if we look at a frequency chart here. So this is the number of samples. Unfortunately, sorry, the screen sharing thing. I wonder if I can get rid of that. Where can I get rid of it? It won't let me move it. There we go. These are the number of samples that we took. So most things were only found in one sample on the port. Okay, so out of all of these samples in here, most of the OTUs were only found in one sample, but the odd thing was found in many, many samples. Okay, but what we can see is large numbers of flies. Not surprising, flies fly and they are attracted to light. So not surprising, we see a lot of those. Coleoptera, we get quite a few, again, flying beetles attracted to light, and moths, etc. So we're kind of seeing what we would expect to see, but the question is, are we finding any pest species? So to get our reference database together at this point in time, what we did was we collated a couple of databases that are available. We got the available species known to be in New Zealand through the New Zealand Organisms Register. And we got all of the names of the species that are on MPI's official pest register. And then we got those together, and then we went out to various international sequence databases like BOLD and Midori, because we have a little bit more confidence in the robustness of the sequence that are submitted to those databases. And we said, well, how many do we have a match for that match the species that we're interested in? So we got together our database that way. And the question is, do any of these OTUs match with things that we know to be around? And more importantly, do they match to any pest species? And indeed, they do. So here's one of the examples. This little thing, Dendhymus versicolor, is a little, seed feeding, plant feeding bug from Australia. And it is in the official New Zealand pest register as a potential pest. So like, oh, okay, maybe we found one of these in our samples, and we're still yet to go back and see if we can physically find these in our samples. But where you see the same reference database, this is our database that we've pulled together for sequences from Midori and BOLD, where we have better confidence that the sequence is what we think it is. Many of you will have heard of or have used GenBank. GenBank, we're aware, has a lot of erroneous sequences in it. And if we were to put this particular sequence that matched in our reference database to this, which is OTU number 273, and we found 160 sequences of that, and it was in 2 of our samples, in GenBank, that also matches to the good old honeybee. So the question is, whose sequence is the right sequence? Is it really Dendhymus versicolor, or is it actually just a sequence of honeybee? Or did the people who sequenced honeybee get it incorrect? So you can see the importance of having a robust sequence library to support your ability to identify what those pests are. And we have a number of these particular hits that we have there. And in some of them, we have actually found hits across multiple ones of our gene regions. So we're a little bit more confident that those might actually be hits on potential pests that we were unaware of. But we're still following through with that. So I just want to finish off by coming back to another element of the reference database. So the reference database is absolutely crucial for implementation of this technology. Everything I showed you in that diagram of things like sieving the samples, extracting the DNA, doing the PCR analyses, and doing the meta barcoding of getting the sequences, that is all very standardized laboratory techniques which people are using around the world, and we use in New Zealand. That's what Wilderlab does. That is their business in Wellington, is providing those sequences. The crux of making it useful for biosecurity is actually having that reference database and being able to separate out New Zealand sequences of what we know to be present in New Zealand with international pest species. So reference databases, we don't have one for New Zealand. And in Australia, they are developing one, and we are scoping out what one might look like in New Zealand currently as another project within Bioheritage. So this project started late last year, and it's known as a Strategic Investment Platform, a SIP, and it has been led by Holden Hohaia and Manpreet Dami from Landcare Research. And they are going through a process right now. And if anyone's interested in this and hasn't become aware of it, I think a lot of people might be aware of it. One of their more recent webinars that they ran to have discussions around this, they capped the registrants at 500 because that's just what the default is for a Zoom room or something. And people were screaming the doors down trying to get in. So lots of people were very interested in this particular topic, is what should a reference library look like in New Zealand? Because New Zealand has, the treaty as an engagement framework, and there is Y262 and the tribunal report, and you can read about that at y262.nz. So the Y262 considers that DNA associated with species is also taonga. Okay, so Holden and Manpreet are going through a process at the moment of what would a national reference library look like that upholds the principles of the treaty and of partnership and of co-governance and co-decision making. And that's everything from the collecting of the samples through to how a functional database may be used at the end. And you may wonder why this is important. Effectively, we could, you could go out there and you could collect sequences and you could create your reference database. And indeed, there is vast amounts of molecular data being created. One estimate is that in the next decade, there will be 40 exabytes of data, of genomic sequence data produced. And to put that into context, 1 exabyte is 100 million gigabytes of data. It's just a phenomenal amount of data that's out there. And what we know about access to such data is if you just make it open, you're just going to embed in place the existing inequities that might be already present in the system. And so we're wanting to avoid that process by going through a co-design process about what should a national database look like. So as I say, if you're interested in that, there's a process going on about it at the moment. And if you are not already in touch with Manpreet or Holden and you want to be, just send me an email or through to Brenda with the registration of this, and we can hook you into that. So the aim of the project that's happening at the moment around the reference database is to engage with tangata whenua, engage with data sovereignty experts, and engage with the people who are kind of the technical people doing the sequencing technology at the moment in New Zealand about what would be best practices for having this reference database. And the plan is by June next year is to deliver a white paper to government, particularly targeting MFE, DOC, and MPI, who would be the major beneficiaries of a national reference database, with what would a framework or a structure for this look like, how could it be operated, and what would it cost to build and maintain that going forward, with the hope that we might get the investment that's required to create it, because the investment required will be significant. Australia is budgeting for theirs something like 40 million Australian dollars. Whether or not they can do it for 40 million Australian dollars, I don't know. So just to summarize from what I've been saying, biosecurity is not zero risk. The incursions are likely to come into various places where trade is related, and we need these tools to protect the things that we value. Okay, and so for the molecular point of view, it's going to be absolutely important to have this national database, and it must be treaty-led. But we're wanting to implement it for a biosecurity system. So it's an interesting conundrum for us. We have to get our house in order for a reference library, but we also need to help others in other countries to get their houses in order, because we will be reliant, since we're interested in pest species coming from overseas, we will be reliant on being able to use their DNA sequences when we find things. If we get our entire house in order, and we know everything that's in New Zealand, and that's aspirational that we could do that in our lifetimes, we will know when an oddity turns up, but we won't know what it is if we don't have access to other people. So international collaboration in this area for DNA-based diagnostics is going to be critical.